After 276 failed attempts and one astonishing success, Dolly, the so-called miracle clone, was created. Dolly marked a historical achievement for genetic engineering, created by a process called somatic cell nuclear transfer. Despite her groundbreaking creation, Dolly's life was cut short, estimated to be around half her expected lifespan. Good morning, I'm Quinn Gorman.
My name is Krish Kapoor.
My name is John Jahan.
My name is Yatvi Gudal.
Dolly the sheep was a prime example of genetic engineering, as it involved the manipulation of DNA to create a genetically identical fused cell. However, since Dolly the sheep lived to half the life expectancy of the normal average sheep, it raises the concern that genetic engineering may raise significant negative repercussions. This leads us to a question. In the past 2 decades, to what extent have the drawbacks of genetic engineering outweighed its benefits? We argue that currently the risks genetic engineering pose overcome its potential benefits to society. This is because genetic engineering has negatively impacted the society through the ethical concerns, the ecosystem, and the economy.
Human cloning is a big debate when it comes to the complications of genetic engineering, as there's more on the line for the participants than anyone else. While considering the perspectives of researchers and participants, it's clear that the ethical concerns outweigh the scientific challenges. As researcher Erica Johnlin states, half of the research anticipated benefits for the participants, yet the consent forms remain vague and ambiguous about these potential advantages, even when optimism wasn't conveyed directly. Patients incorrectly make the assumption that these genetic engineering trials will result in positive outcomes due to the ambiguity of the consent forms they sign. Success is not guaranteed in these trials, so to make consent forms clear, we must mitigate this ethical concern.
In such human trials, it can also be seen that gene editing may become ineffective. Antibodies are proteins that are naturally produced by the immune system to defend against foreign antigens such as bacteria or viruses. Natural antibodies may see gene editing tools as foreign material, which results in the human body's immune system attacking against these gene editing tools. One of these tools is the CRISPR-Cas9 system, which relies on the Cas9 proteins that are produced from the bacteria S. aureus and the bacteria S. pyogenes. According to Andrew Joseph, research conducted by Stanford University found that in a sample of 22 newborns and 12 adults, 79% of the sample contained antibodies to the bacteria S. aureus, and 65% of the sample contained antibodies to the bacteria pyogenes. Clearly, this shows that the human body recognizes these bacteria as foreign material, showing that the Cas9 proteins are inhibited and making the CRISPR-Cas9 system ineffective and not a feasible treatment option for patients. In addition, the concerns can be extended beyond humans and shown among a variety of organisms. Telomeres are protective caps at the end of chromosomes that become shorter as cells divide, until they lead to cell death known as apoptosis. According to research conducted by Ji Zhu at the University of Connecticut, the terminal restriction fragment size, which is a measure of telomere length, showed a statistically significant decrease in the mammary gland, embryonic, epithelial, and oviduct cloned cells when compared to their controls. Clearly, this shows that telomere shortening seems to intensify in genetic cloning, which shows, since telomere lengths are indicative of cellular aging, that if these lengths prematurely shorten, the lifespan of organisms will significantly lessen, showing yet another repercussion of genetic engineering.
In this graph, you can see the representation of survival rates of calves after a subsequent experience genetically modifying the offspring of cattle. This experiment produced a total of 22 fetuses and quickly dwindled over a 90-day time span. Moments after the experiment, 5 cattle had died. An hour after, 3 more had passed. A day, 2 more, 21 days, 2 additional, and finally, at the end of the time span, 1 more cattle had passed away. This totals the death to 13 deaths, which is a 55% decrease from the original pool of calves. This shows the moderate failure and ethical concerns that genetic engineering possesses.
However, the death of some organisms may not only occur through intentional DNA manipulation. Sometimes it can occur through side effects. As stated by David Pimentel, a professor at Cornell University, he states that the pollen from BT corn is confirmed to be toxic to the remarkable monarch butterfly, already threatened by its naturally wintering sites. As depicted in this graph, the overall downward trend from 1997 to 2016, 1997 being 682 million, 2016 being 150 million, is a result of the increased use of BT corn. BT corn was first implemented in 1996 and was specifically designed to kill off pests that feed off the corn, specifically the European corn borer. However, milkweed, the plants monarchs feed off of, tend to absorb the toxin used to kill the borer. Since BT corn and milkweed are around the same area monarchs resided, this has caused a decrease in population.
However, as you can see in this graph by Kim Anderson and Chantal Nielsen, it shows the global agricultural area of genetically engineered crops. In 1996, there was roughly 12,000 square miles of genetically engineered crops globally. In 1999, there was roughly 156,000 square miles of genetically engineered crops globally. This is a 13.3 times increase from 1996 to 1999. This land was used to show how economically we grew globally. Despite this economic benefit, jobs evidence shows the monarch population in millions. As we go to the time period, 1996 through 1999, it was at 700 million, it was at its peak in 1997. And it drastically dropped from 1999 to 200 million monarchs in population, suggesting that the increased use for GMOs is harming the monarchs' butterflies, which is a clear concern to the economic benefit. Additionally, cross-contamination is a fear of many farmers across the globe. Biotechnology crops have the potential to crossbreed with other plants, resulting in many drastic breeds. Consequences for biodiversity are far more severe than a simple economic inconvenience. In this image by Tor Jansen, it shows the blue corn crossbreeding with the yellow corn. This crossbreed shows crossbreeding. If this contamination shows crossbreeding. If these plants cannot be monitored, a more extreme remedy such as a moratorium or ban may be put in place in order to save the crops, which would hurt the farmers' profits.
However, cross-contamination may not only occur through crossbreeding, it can also occur through herbicide mutations. As stated by Carol Hoffman, a genomic science specialist in Massachusetts, she states that herbicides considered environmentally safe would no longer be effective against weeds that have captured the gene for herbicide resistance, forcing the use of more dangerous chemicals. As stated previously, the CRISPR-Cas9 system is a gene editing tool. This tool allows farmers to alter the traits of which they want to enhance or inhibit, such as crop yields or taste. As depicted in the diagram by Hyung Dong, once a CRISPR-Cas system is in play in the plants, mutations may occur. Once the farmer starts spraying herbicides repeatedly over the plants, herbicides will start building natural resistance towards them, prompting farmers to use stronger herbicides, causing more damage to the environment than good. Overall, however, current research suggests that genetic engineering poses ethical, ecological, and economic concerns that counteract its potential benefits to society. As seen previously, genetic engineering has posed issues with informed consent, showed clear trends of reduced lifespan in infected organisms, and detrimental effects on agricultural profits. With additional research, however, genetic engineering may prove to be feasible in disease prevention by altering genes responsible for the spread and development of them, increased crop yields. GMOs already show increased profits that benefit the economy. Although currently the harm GMOs inflict on the ecosystem outweighs economic benefits, if future research mitigates these consequences, the benefits of genetic engineering may overcome its negatives. And finally, safety and efficacy. By conducting more controlled trials, current side effects of gene editing and cloning can be mitigated, making it a viable treatment option for patients. Thank you.
All right, we'll just start here and move our way across. Chris, can you tell me how you decided to use Anthony's paper in your presentation?
We decided to use Anthony's perspective since his research was mainly positive. It showed, we used it as a counterclaim that showed how the positives may outweigh the negatives. However, we used it as a counterclaim, and since the rest of our peers was primarily negative, we showed how this economic benefit, such as the crop yields he mentioned to increase farmer profits, was outweighed by the negatives it had on the environment.
All right. Job, tell me, reflecting on your colleagues' work, which one had the greatest impact on your understanding of this issue?
I believe Kirsch's paper had the greatest impact in understanding this issue, as Kirsch's paper served as the scientific basis of genetic engineering and has allowed me to see why genetic engineering has such a toll on the environment and how I can formulate my argument based upon that.
Okay. Sorry. Anthony, What's an example of a compelling argument from one of your peers' reports that you guys didn't use, and why didn't you use it?
An example of a compelling argument we excluded was Quinn's ethical part about religion, mainly because it didn't tie into the presentation and it just didn't really connect well with the flow of the presentation and how we were going to present it.
Sure. And Quinn, if you had had a fifth person on this team, what could they have brought to this that would have helped?
I think if we had a fifth person on our team, a perspective such as political could be really beneficial, as you could incorporate government law and their policies against genetic engineering, and I think that would flow really well with my ethical perspective and Anthony's economic perspective.
You guys are done.